So to basically summarize my hypothesis, it is essentially a lab origin in the U.S. and a lab leak in Wuhan based upon that simple evidence of the transmission model of this virus.
Welcome, and thanks for joining us. All truth passes through three stages. First, it is ridiculed. Second, it is violently opposed. Third, it is accepted as self-evident. Arthur Schopenhauer. Where did SARS-CoV-2, the virus responsible for the COVID-19 pandemic that ravaged the world, come from? No matter what you may think, the origin of SARS-CoV-2 remains unknown. Why is it important to determine the origin of COVID-19? Knowing how a pandemic started is the key to preventing the next one. Understanding how SARS-CoV-2 entered the human reservoir will help us prevent a similar pandemic in the future. We need to find the truth, no matter how painful that truth may be. Two competing theories with vastly different implications quickly emerged to explain how a novel bat coronavirus entered the human population in Wuhan. The first theory postulates a novel bat coronavirus jumped from an animal species into a human in what is known as a natural spillover event. Of note, in the 4 years since SARS-CoV-2 infected humans in Wuhan, China, no animal reservoir has been found in China to explain the spillover, despite an intensive search for that reservoir animal, and despite numerous attempts to juke the genomic and epidemiological data by self-interested virologists to make it appear that COVID-19 was the result of a natural spillover. The second theory proposes that a novel SARS-like coronavirus manipulated in a lab escaped from that lab, most likely in an infected lab worker, and entered the human population in Wuhan. Of note, in the 4 years since SARS-CoV-2 infected humans in Wuhan, there have been 0 forensic investigations. 0. And that, despite knowing that 4 investigators funded by U.S. tax dollars had proposed research that has been characterized as, quote, the blueprint for creating SARS-CoV-2, end quote. In the last episode on the origin of COVID-19, microbiologist Valentin Brutel presented abundant and compelling molecular evidence that points to SARS-CoV-2 being an engineered virus. Most notably, the furin cleavage site with a well-characterized restriction site adjacent to it, perfectly situated to split the S1 and S2 subunit of the virus to facilitate entry into the cell. No sarbecovirus in nature except SARS-CoV-2 has a furin cleavage site. For years, virologists have known that a furin cleavage site strategically located in the spike protein can make a virus more pathogenic and more transmissible. It can also make the virus capable of crossing species barriers. Many experts studying the body of molecular and circumstantial evidence believe the preponderance of evidence points to SARS-CoV-2 being a lab-engineered virus that inadvertently escaped into the human population in Wuhan. What we do know is that there was a great deal of research being done on SARS-like coronaviruses in Wuhan for years in collaboration with U.S. scientists at U.S. institutions funded by the U.S. government, namely the NIH, the NIAID, and USAID. Much of this U.S.-China collaborative SARS coronavirus research was coordinated by Peter Daszak of EcoHealth Alliance, subcontracted to Zhengli Shi at the Wuhan Institute of Virology, Ralph Barrick at the University of North Carolina, and Linfa Wang at Duke University in Singapore. We know that China isn't sharing any data, but what fails to receive much attention in the mainstream media is that 4 years later, the NIH, the NIAID, USAID, Ralph Barrick, and Vincent Munster at Rocky Mountain Laboratories have also refused to disclose any details of their SARS-related coronavirus research in the 2 years leading up to the COVID-19 outbreak in Wuhan. When the NIH was finally required by a Freedom of Information Act lawsuit to turn over all coronavirus research they funded, 2 years after the initial request, they turned over 292 redacted pages to The Intercept. So, if this isn't a cover-up, why on earth wouldn't they tell us what research was being funded by the NIH and the NIAID? In the Project Diffuse proposal, Peter Daszak writes on page 23 that EcoHealth Alliance will, quote, maintain a central database of data collected and generated via all field, laboratory, and modeling work, end quote. So, we know there are multiple copies of all SARS-related coronavirus research being done in the lead-up to the COVID-19 outbreak in Wuhan. And yet, no one has produced those data and records for independent scientific and forensic analysis. And no one in government has demanded they turn over all those data and records. And last week, we learned from a CIA whistleblower that 6 out of 7 analysts who initially opined that a lab leak was more likely than a natural spillover were either coerced or enticed financially to change their opinion from lab origin more likely to we can't tell where this thing came from. And we know that Drs. Fauci, Collins, Farrar, and the Fab 4 evolutionary virologists authoring the Proximal Origins correspondence misled the trusting public to believe that they had shown that SARS coronavirus 2 was not a laboratory construct or purposefully manipulated virus, while contemporaneously exchanging private electronic messages that they still believed the lab manipulation of the virus was quite possible, if not likely. We can no longer trust virologists to speak the unvarnished truth about what they believe regarding the origin of COVID-19, even at the highest levels. Nor can we trust our intelligence agencies or law enforcement to conduct a thorough investigation into the origin of COVID-19. They've had 4 years to demand that the NIH, NIAID, USAID, Rocky Mountain Lab, and Vincent Munster, and University of North Carolina and Ralph Barrick to turn over their lab books, their research, and any and all data related to SARS-like coronavirus research in the lead-up to the COVID-19 outbreak, to conduct a proper investigation. But they have not done so, which again speaks volumes to a concerted cover-up. And there's no reason to believe they'd cover it up if they believed they could hang it on China alone. So the origin of COVID-19 will not be determined by virologists looking at genome sequences or phylogenies. They have already shown us they cannot be trusted to tell us the truth. And they also have a massive financial and professional conflict of interest. To determine the origin of COVID-19 requires a forensic investigation, like a homicide investigation. But unless and until they conduct a forensic investigation into the origin of COVID-19, it will remain a decentralized investigation. Which brings us to today's episode. If you recall from the episode on Project Diffuse, the central goal of that proposal was diffusing the threat of spillover of bat-borne coronaviruses to humans. The explicit purpose of Project Diffuse was to immunize bats in caves with engineered viral vaccines and other immunogens to reduce the risk of SARS-like coronavirus spillover to humans. But DARPA never funded Project Diffuse, you say. That's true, but based on the written testimony of Major Joe Murphy, Office of Naval Research, and former DARPA fellow, DARPA did reject the proposal because the work was too close to violating the gain-of-function moratorium. But he claims that Dr. Fauci did not reject the proposal and that the work took place at the Wuhan Institute of Virology and several sites in the U.S. In short, Murphy claimed that SARS coronavirus 2 is an American-created recombinant, self-spreading viral vaccine intended for bats. Sound crazy? Of course it does. Like a lot of gain-of-function research, it sounds wacko at first. Self-spreading viral vaccines were a hot topic in 2018 and 2019. Infectious vaccines theoretically can be administered to humans or animals. A self-spreading vaccine is a live virus capable of infecting a target reservoir animal like a bat. But instead of causing disease, it confers immunity. Why create a self-spreading vaccine? You can't immunize bats one at a time. And even if you could, you'd never be able to reach anything like herd immunity in a colony of 10,000 bats. So instead, create a self-spreading vaccine and infect a small proportion of the bats in the colony, called the founder bats. These founder bats are then released back into the caves where they originated, where they can transmit the infectious vaccine to the rest of the colony. In this case, the self-spreading vaccine would immunize bats to SARS-like coronaviruses that present high risk for human spillover, like SARS-1. Instead of spreading disease, the vaccine virus induces herd immunity against SARS in the entire bat colony. Theoretically, this herd immunity makes the bats less likely to acquire SARS-like viruses, and therefore less likely to transmit them to infect other animals outside the bat cave, including humans. Before you decide that this theory sounds too crazy to be true, suspend judgment until you've heard the entire case by today's guest, Jim Haslam. Jim has been investigating the origin of COVID-19 for 3 years and has studied the available evidence in the public domain. He presents the evidence for his self-spreading bat vaccine hypothesis in a series of essays on Substack. I've included links to Jim's Substack on the Straight Talk MD website. Today, we discuss his hypothesis and the evidence supporting it. I'm Frank Sweeney, and this is Straight Talk MD.
It is a system which has conscripted vast human and material resources into the building of a tightly knit, highly efficient machine that combines military, diplomatic, intelligence, economic, scientific, and political operations.
The one thing that I found really sort of haunting was that when I read the 6 full essays and you go back and you listen to what people have said, it actually completely changes your perspective. For example, when people were saying there's no way this could have been created in Wuhan, I kept saying these are just a bunch of self-interested virologists who don't want to admit that this could have been engineered in a lab. But their specific language was, this wasn't made in Wuhan. They didn't say it wasn't engineered per se. And this is like the slightly different perspective is if you assume that it wasn't made in Wuhan, but leaked in Wuhan, a lot of this makes a lot more sense. Okay. And so that's the whole point that we're going to go through today. But I want to let the listeners know how detailed your essays are and that they seem to create a very, very, very believable hypothesis.
I have nothing to hide, to be honest with you.
Okay. So I can't promote you as an ex-CIA operative in the intel because... So this is really interesting. So I follow you on Twitter and so on, and the picture on your X, it's X, I keep still call it Twitter. That it looks like some sort of a Romanesque building, and it says something, but it's blurry, and I can't read it. What is that image that you have on your X or Twitter?
It's something that's been there since my Twitter account was started, which I'm not even sure when it was, at least a decade ago.
Okay.
So It's Cairo. I'm in Cairo.
Oh.
And I'm in Cairo between regime changes. So I'm not there as some CIA op. I'm there as basically a traveler, a solo traveler in between European jobs. So I basically flew down to Cairo, and where else did I go? I have to think about that trip. Israel, Jerusalem, and all that.
This is definitely sounding more like some intel background, but
I know.
I'm not going to probe it. I'm not going to probe
You're the
it.
second person to ask me about that picture, but yeah, it's the university, or it's I don't remember what it was. It was the American University in Cairo. I think that's what it is if you look closely. So yeah, if people see that image and they see my last name Haslam, and it's Haslam, it's British, and they get a little confused. But no, it's really not much to it. It was just Cairo late at night. Wow, look at this. There's an American University in Cairo.
Okay, so that's what I thought I saw. I thought I saw an American University, and I started going through images for the American University. I never got to Cairo, so that's why I never saw it. I was looking to try to match it up to an American University because that's what I thought it said, kind of blurry.
Yeah.
How'd you get started in this? I know you're in the intelligence industry, but how did you get started in this investigation?
So my story is probably like everyone's story. I'm minding my own business in January and February of 2020. And this, mysterious pneumonia emerges in this mysterious town that I didn't know about, and I bet you none of you knew about, called Wuhan. Even though it's a city of 10 million and it's huge, I knew nothing of it. So I'm carrying on with my life like everyone else. And the first inkling I had that something was going, weird was when Zero Hedge had their Twitter account banned for reporting that there was a BSL-4 in Wuhan. And I was like, well, I can put two and two together. I don't really need to be a rocket scientist to figure this out. Something happened. And I'm going to move off my life because I don't think any of us could ever prove it. There's probably records that have been incinerated and blown up and done away with, and it's China. You're not going to get in there and figure out what happened. So I was, it's just the flu, bruh. Let's move on with our life. And, as the thing spread around the world in rocket-like pace, I mean, within weeks, basically, it got from Wuhan to America. And I'm like, wow, this is something else. But I'm still of the opinion it's just the flu, bruh. And I'm kind of the mad opinion that, the mask and the lockdowns and all the other stuff that came along with it was just completely unnecessary because I'm now of the mindset it's Chinese junk. It's going to burn out, before the end of the year. And I have moved on with my life, Frank, and I have, basically, I'm a podcast listener, and I listen to your podcast, and I'm basically doing a road trip at the end of 2020, and I'm listening to the Joe Rogan podcast. It tells you what kind of guy I am. I am not intel. I am, I am a literal, nobody, and I plan to remain a nobody. I'm a proud nobody. If I'm not, talking about this, I'd probably be on the couch getting ready for, college football. So it's basically, in the end of 2020, it comes roaring back. And I'm like, well, that's funny because that just completely debunked my hypothesis that it leaked from the lab without doing any investigation, without doing any, research on it, and assuming it was Chinese junk. And it came roaring back at the end of 2020. And on the podcast of Joe Rogan was Alex Berenson. He's somewhat infamous, but he wasn't talking about what's he talking about usually. He talked about the peculiar furin cleavage site. That was the term he used. And he said the French found it peculiar. And I go, well, I find that term peculiar. So I slowly started looking into the genome sequence at the end of 2020. By this time, everyone's online doing their own, research. Everyone knows about, gain of function, Ralph Barrick, Peter Daszak, and all these others. I don't. Then beginning of 2021 was Nicholson Baker's New York Magazine article. And in there, he mentioned these two white guys 60 times, Peter Daszak and Ralph Barrick. It's basically Peter Daszak of EcoHealth in New York City and Ralph Barrick of the University of North Carolina. And he mentions them 60 times with great detail and great research. And I'm like, well, there must be something here. But I don't know quite what it is because both of them have been quite open and honest about what they did and didn't do. And it obviously came from Wuhan. It didn't come from America. So you really just can't put two and two together. So to make a long story short, I'm sitting there kind of banging my head against the wall. What on earth is going on here? Nothing makes sense. And, I slowly started to put the pieces together once a grant called DARPA Diffuse leaked. So that's kind of my story in a nutshell.
Yeah, the DARPA was really a huge factor in laying out how this could have happened.
Yeah.
The complexity of what you presented in your, this is like 400 pages The detail is incredible. I mean, and the way this thing was put together with all the references. So I'm not just talking about sound clips from Fauci talking or Barrick talking. They're in there also. But all the papers, so when you make a claim or a statement, there are several references in there, and I read every one of them to say, okay, does this really say what he says it said? And the answer is yes, it did.
I'll put out my theory in 3 sentences.
Okay, I was actually going to quote your thing. It said the virus, meaning SARS coronavirus 2, was an animal vaccine created in Chapel Hill, North Carolina, developed in Hamilton, Montana, and tested in Wuhan. This is where it leaked accidentally, but the creation and cover-up was not an accident. So go ahead and briefly state your theory.
Well, yeah, I completely forgot about that line, which is better than the one I was going to give you, Frank. So, yeah.
I've been candid with you. I have been trying for 2 weeks to break your theories and what you say in your 6 or 7, essay length pieces. That this could be a book, by the way. It's a tome. But I've tried to break any part of it down, and I can't. And this has been bothersome to me. So what I don't understand is everything I read in your essays seems to comport with the lab leak theory. It's just your hypothesis says created in the U.S., being tested in Wuhan in a BSL-4 lab, and it jumped there, a lab-acquired infection most likely from while they were testing this. We're going to talk about self-spreading vaccines. That to me is not some big difference in the 30,000 foot, how this might have happened. It seems this creates a lot of attention on Twitter or X between you and lab leakers. What's that about? I don't understand why they can't believe this is possible.
So I was with them, shoulder to shoulder up until August of 2021. I am basically, I'm really getting into it in the late summer because I'm still banging my head against the wall, and I'm looking for answers, and I'm reading probably about 2 to 3 hours per day over the past 2 to 3 years is how I was eventually put all this together. But in August of 2021, I'm basically, in lockstep with all the other quote unquote lab leakers that, yeah, this is, some sort of PLA military agent that got out accidentally. I've always been of the opinion it was an accident. I mean, no one, intentionally releases anything in Wuhan that ties back to the doorsteps of Beijing and Washington, D.C. So that would be ludicrous. But it was always an accident in my mind. And then I'm having the opinion that it's now got to be a military funded project because it spreads so easily. It floats through the air upwards of 60 feet. And I'm looking into, various PLA officers that can, do aerosol work. And I'm basically diving deep into PLA research in August of 2021. So these aerosol officers aren't really in Wuhan. They're over there by the North Korean border. I am really getting into this. I am with them. Essentially, lab leakers, as we would call them in general. Mm-hmm. It leaked from Wuhan. It was made in Wuhan. It's Chinese, work, and it's Chinese fault. So I basically was with them. And then in August or September, I guess, of 2021, a DARPA diffuse leaked. And the Drastic people, who I'm not a member of, never was, never will be, posted it to their credit, to their defense. And they posted it, under the assumption it wasn't funded, even though what it was inside the grant was obviously something that was going to be created inside the United States, then shipped to Wuhan for testing. And I'm of the opinion, well, everything's funded when it comes to Washington, D.C. This completely 180s my view. And after I read through it the first time, and there in there is the infamous line, we will insert a fear and cleavage site into live coronaviruses and test them on live bats in Wuhan. 2 weeks after that, after a couple of long walks, I come back and I go, well, this is what did it. Mm-hmm. And so I went on that hypothesis it did. And that's where I started going in a completely different angle than everyone else, because I went on the assumption it was funded, knowing how Washington, D.C. works. Everything's funded.
Okay, a couple things there. Number one is everybody who read that diffuse proposal knew that kind of research was being done at the University of North Carolina. Ralph Barrick had been putting fear and cleavage sites in SARS-like viruses already. And Ralph Barrick had obviously the largest technological capability in reverse genetic engineering and the largest lab of anybody in the world. So is their main beef it couldn't have been created there? It had to be created in the Wuhan Institute of Virology. No one believes that any more than to say that the nuclear bomb detonated on Hiroshima had to have been made in Japan because it detonated in Japan. That's nonsense. The vaccine could be created in the U.S. and shipped to Wuhan, I think you said, in about 48 hours and tested. I'm not saying it's University of North Carolina, but I would say that would be my number one suspect. So you said Diffuse wasn't funded. That's the big argument. And we all know that's the fact that DARPA didn't fund that specific grant doesn't mean it wasn't done. It's nonsense. It's complete nonsense. Yeah. But the other thing that you passed on to me, I got all the documents from Major Joe Murphy. He's the guy who originally released that Diffuse proposal. That's the one where Major Joe Murphy said, I'll read you the exact line. He said, DARPA rejected the proposal because the work was too close to violating the gain-of-function moratorium, despite what Peter Daszak says in the proposal, that the work would not. As is known, Dr. Fauci with the NIAID did not reject the proposal. What did he mean by that? He did not reject the proposal. He implies that while DARPA rejected that proposal, Dr. Fauci at NIAID funded it.
Yeah, well, that's basically what he is saying by, being clever with it. So Anthony Fauci's been at the head of NIAID for at least 40 years. He's been at NIAID for 50. We all know his story by now. I don't know if I knew of him before COVID. I may have. I pay attention to the news. I knew of the HIV outbreak and his being the face of that one. But in 2002 or 3, after 9/11, Fauci's job description changed. And if you go in there, and that's part of the references I link, and going back to that point, that's why I reference so much, because I'm, again, a nobody, and I want to basically have the references do the talking, not me. So in 2002 or 3, Fauci's job description changes, and he's basically now the head of the U.S. government biodefense agency, if you will, which is now NIAID. And if you listen to some, it was a bit of a workaround because the DOD couldn't do it legally, but NIAID could do it. What is it? Gain of function, or eventually became gain of function. So it is in Anthony Fauci's job description to fund something like DARPA Diffuse. Now, a lot of people are going to say that's crazy because DARPA Diffuse is crazy. Well, we know it's crazy, post hoc type thing. It basically, pre, lab leak, it doesn't look that crazy. It is an idea to prevent a natural zoonosis spillover event before it occurs. It is grandiose. It is, something DARPA would come up with and something that NIAID would fund because NIAID's agency is twice the size of DARPA. So if anything, Murphy basically told us outsiders what was going on the inside, and that Fauci funded a revised version of DARPA Diffuse, which is what I basically hang my hypothesis on. So if you look at the losing team of DARPA Diffuse, it's Peter Daszak, it's Ralph Barrick, it's Linfa Wang, it's Shizeng Li, and it's basically a group of people. Those 4 people have been working with Fauci for 10 years? In the open
or more, yeah.
So this is a tight group. They have done some, I would call it great work, in their mind because it's being funded with great deals of money by Anthony Fauci. And by that, I mean the R01 work, which everyone alludes to. The hypothesis that lab leakers use is that Fauci kicked some of your tax dollars over the Pacific Ocean into Wuhan, where an accident occurred, where Shizeng Li tried to cover it up. And that's the leading hypothesis pushed by most, sane lab leakers. It does check a lot of boxes, but when you dive down deep into it, then none of the boxes are checked. So to segue into, what I think I really want to get into everyone's mind is that after 3 years, lab leak, as we will call them, I'm not going to name names, but we know who they are. They were me, a year or two ago, have had 3 years to come up with one piece of biological evidence that the Wuhan Institute of Virology created SARS-2, and they have none. Zero. Zilch. Nada. I know. I've looked. I've asked. I've yet to find any. They have tons of geographical evidence. Obviously, the Wuhan Institute of Virology being, a few miles away from the epicenter of this outbreak is quite coincidental and not hard to, see through. But when it comes to biological evidence, they have none. When you dive down into the details of what they're proposing, none of it held up to scrutiny.
if we're assuming that this thing was engineered, and I believe to a very high, after talking to experts in the field and going into molecular biology that is, mind-numbing, there's a lot of molecular evidence that this was engineered. So in Project Diffuse, you said one of your assumptions was that Diffuse or something like it was funded, and this research was done. But if this was done, this was designed to diffuse the threat of bat-borne coronaviruses in China, and this was around 2018. And their big push was broad-scale immune boosting versus targeted immune boosting. The targeted immune boosting would include things like self-spreading vaccines and so on, the one that we think this likely could be. But the technology required to do that was all going to be done at the University of North Carolina. And that's the one thing that was clear over and over again in the Diffuse proposal, was that the reverse genetics to create backbones and to put in different spike proteins and all that was never going to be done in Wuhan Institute of Virology. Ralph Baric himself said that, and you pointed that out. He said that. You actually have clips where he himself said, we didn't share any of this, this technology with the Wuhan Institute of Virology. This was all done here. We didn't even share that with them. So all the high-end technology that needed to be done had to be done at the University of North Carolina. That was the only place that had all the moving parts to do this. And supporting your argument, there were a whole bunch of people that their interpretation, how I interpret them, changed after looking at this. Even Christian Anderson said he didn't think that this could be done at the Wuhan Institute of Virology, but he certainly did believe it could be done at the University of North Carolina. And Christian Drosten kind of rejected the whole Wuhan Institute of Virology engineering idea because he believed they did not have the technical capabilities to create things like self-spreading vaccines, self-spreading SARS-like vaccines, and doing all the reverse genetics. These experts who kept saying, you're crazy, it couldn't have been synthesized at the Wuhan Institute of Virology. What they never said was that it couldn't be done at the University of North Carolina and sent for testing to the Wuhan Institute of Virology, which is the way I interpret, that's your basic theory, and very believable, is that all the technical creation of synthetic viruses, consensus genomes and things like that, all could be done at the University of North Carolina. And that is consistent kind of with all the people at the February 1st teleconference with Fauci. They all didn't invite Ralph Baric to that meeting because they believed he was the most likely person who was involved in the engineering of this virus. That was their claim afterwards. They said, well, the reason we didn't invite Baric, who's the smartest guy in the world on this, to our meeting is because we thought he was probably involved in the engineering phase of it. So they said that outright right there. So how were they planning on testing these self-replicating virus vaccines? So basically, you take a vaccine, you're going to infect the bats with it to make them immune to all those SARS coronaviruses so they don't infect us. But you're going to do it in their caves. This is the part where you have a great deal of knowledge how they carried that part out. Why don't you go into that?
So there's always been a dark cloud over Ralph Baric at the University of North Carolina, but we've never been able to connect him to Wuhan until the DARPA Diffuse grant leak, where he literally said he would insert the virion cleavage site into live SARS-like coronaviruses and test them on live bats. He's going to lead. It's in the DARPA Diffuse proposal. He's going to lead that section of the grant, a $14 million grant. And what Baric proposed in DARPA Diffuse, in his defense, was perfectly safe. It is ground-based. It's not transmissible. It goes into a humanized mouse model, which doesn't transmit SARS-like diseases. It can't even infect us humans. We have a test from that because 10 people inside of his lab have been exposed to infected mice with coronaviruses, and none of those 10 were ever sickened. So they're never in the history of coronavirus research has a mouse ever infected a human and spread around the world in weeks. So everything Ralph Baric did in his own defense from 2000 to 2019 was safe, ground-based, non-transmissible. I was always suspicious of Ralph Barrett because when he was asked about transmissible research, he got very uncomfortable, and I thought he was trying to hide something. If you go deep into my Twitter feed, you can basically see me thinking that he defected to the PLA, because it's always been in his lab. His technology is his lab. He's not giving away his technology to the Chinese. He's got a $100 million lab. He just doesn't give it away. This is knowledge based upon, a lifetime of work. He studied over there in the West Coast by you under Michael Lai, who is the father of coronaviruses. So he has a lifetime of studying this kind of research. And it's only him on all the papers, basically, all the pertinent papers, along with one of his 30-year bench scientists, Boyd Yunt. So, but everything he did from 2019 is safe, ground-based, not transmissible. Even the DARPA fuse proposal, as it stands, is safe. A fear and cleavage site in a coronavirus inside a humanized mouse cannot infect you. It could bite me. I could eat it. It's not going to infect me. It's not going to spread around the world in weeks.
By the way, in support of that comment, you actually had a clip in there where Ralph Barrett himself said that these things don't cough and sneeze, so they don't transmit this virus. Barrett said that.
Correct, and that's the part I try to hang on. But if you go to the second line of that statement, it's even better. As you adapt the virus to a mouse, it actually moves away from its ability to infect a human. So it is safe. The guy has been able to push, Ralph Barrett has been able to push the genome sequencing of these viruses to the bleeding edge of just an insane amount of technology because he has a safe model to test it in. So he does what's called a no-seum sequencing. And a lot of people are, why is a university professor doing no-seum research on reverse genetics for coronaviruses? And there's a moral reason for that. Because when you're trying to create an animal vaccine, you need to hide the engineering scars from the animal. You have to trick the animal's immune system into thinking the virus is natural. And therefore, the immune system accepts it and then produces antibodies against it. Now, if I come in and try to engineer something like Xizeng Li would, and it has all these little scars and tissues, and it looks like Chinese junk, and I try to infect a Chinese bat with it, I'm not going to be able to do it, nor would she. But if Ralph Barrett at the University of North Carolina went to create a genome from scratch, what's called a consensus sequence, as he put it inside DARPA Diffuse, to be able to trick the animal's immune system into thinking the virus is natural, then you could create an antibody response. Then you could create the animal vaccine, which is what he's all about. If there's anything that we all agree about these virologists, they want to vaccinate every living organism on the planet, not just us humans. We kick and scream as we learn, or quite a few of us at least, when it comes to the vaccination. But when it comes to animals, there's no, FDA approval. I mean, it's like 2 or 3 years for an animal vaccine. It's 20 years for a human vaccine. And you can't really test on human subjects anymore. Monkeys are expensive. So, back to where we started here, we're trying to find a transmission model for this darn virus. And it's not easy to do in the first year or two because this thing hasn't sorted itself out. So Ralph Barrett, in that same interview in what was it? I'll say May or June of 2021 before DARPA Diffuse Leak. He was pretty honest. He said, I never sent genome to China, period. And these people speak in half-truths, white lies. They are brilliant people that can basically not get caught in a lie, per se. And I took that for, gospel. And I filed that away. And after DARPA Diffuse leaked, I brought it back out. And I'm still looking for the humanized mouse transmission model of SARS-2. And I slowly start getting my brain around it. And once you do, you start to realize that there is no one in North Carolina that could have taken this agent, which is Ralph Barrett's genome sequence, and aerosolized it. There's no bats inside the UNC lab. There's no monkeys inside the UNC lab. Ralph Barrett is on record as being very uncomfortable with transmissible studies. And I start slowly looking towards animals around the Montana region. It's just hard to avoid it. All right.
Okay. Well, but you have to explain why you're saying Montana, because in the original DARPA proposal, it was Tony Rock, and she's with the U.S. Geological Survey, and I think that's in Wisconsin. Her lab, or the lab that she was at. Now you're talking about Hamilton, Montana, and that's really important because that's the 6th Munster. So give a little background on why you said Montana, because we haven't broached that yet.
So just to back up a little further, people need to be comfortable with the fact that viruses created in one spot can be tested on another. There's a great line by a Montana professor who I approached with my theory. I'll come back to that. And his line inside, one of these articles, and I've I've read basically everything. I harvested the internet trying to find this answer, was that the mail is full of plasmid dried filter paper with viruses on it that are sent around the world into other labs. And, this actually was an issue. A Canadian scientist lost her job when she sent some virus samples to Beijing. So these people live in a very small world. These people are virologists. They go to the same conferences. They hang out at the same, conference universities and everything else. So they are a very, small niche group, and they share samples with one another. They collaborate together. So you have to understand that, something created in one place can be tested in another. So what brought me to Montana is, where the story gets a little weird. So in the end of 2021, I have read DARPA Diffuse. I have taken Ralph Baric for his word. These are people of action. I'm going to insert the furin cleavage site into the virus, and I'm going to test it on live bats in Wuhan. And I know that, what Ralph Baric created genome-wise could not be created virus-wise, because this thing is spreading like, wildfire. And then in the end of 2021, there's a virologist on Twitter who's basically talking about the virus in the deer, the American deer. And he doesn't call it antibodies. He calls it virus. He makes the distinction that this is not antibodies showing up in deer, because antibodies show up in basically every animal on the planet, which basically means you stop transmission. Cats, dogs, monkeys, when you show antibodies, you have stopped the transmission. But what's not stopping the transmission in deer is live virus. They did PCR tests on these animals, basically. American scientists went out to the American wild and did PCR tests on these animals. And that just basically makes everyone's brain go in all kinds of different directions. But once you get your brain around the fact that it's not antibodies, it's virus living inside the deer, I go, that is utterly insane. And there ain't no deer inside UNC's lab.
And an important point, that because this distinguishes it from the raccoon dogs who were not infected with the original Wuhan strain. These deer had some of those original viral strains, the earliest viral strains, because as the virus got more virulent but less pathogenic, sometimes the virus may transmit to other animals. But some of these studies, I think, that I read here, were that the earliest strains, the original viral types, were the ones that were infecting them, which means it was very early on. This was an early version of the virus was able to infect these white-tailed deer.
Correct. Yes. It was Brian Hagel, Hgelle. It was his Twitter feed. He is a professional retired virologist living in New Mexico. He's since deleted his Twitter account because I think he went off the edge. But he was the one talking about the fact that this is not antibodies in deer. It is virus, living virus. And when you look at Brian Hagel, he is a virologist who was actually open to the lab leak theory. He pointed his finger at the Wuhan CDC. He wasn't involved in, kind of the nitty gritty, trench warfare that's been going on between the two groups. But he was kind of on the sideline, and I was one of the few that followed him. And I started looking into Brian Hagel, and he not only studied, in the Midwest. I believe he was in was he always at the University of New Mexico? If not, he was always studying what's called deer mice. And then that was the second animal that the virus spilled into. Those are also found along the Rocky Mountain Ridge. So all these animals keep popping up that shouldn't be popping up, essentially. What I call New World animals are showing the ability to not only infect but transmit this virus. Not Old World animals. There's not a single Old World animal, meaning the Eastern Hemisphere, that is shown to transmit SARS-CoV-2. But there are 5 New World animals, I would call them, that are basically showing transmission capabilities of SARS-CoV-2. So to basically summarize my hypothesis, it is essentially a lab origin in the U.S. and a lab leak in Wuhan based upon that simple evidence of the transmission model of this virus. And to get really into the weeds, whenever I approach a virologist, I basically do this in a way that is somewhat professional. But why does lineage A, the ancestral strain, the virgin virus from Wuhan, transmit in American deer mice, in American deer? ? And every virologist kind of knows the answer, because it's really, really, really crazy. But you have to kind of be a full-time virologist to understand it. And they won't answer it. And if they do, your answer would be something like, to close this out, what the University of Montana professor came back at me with. I can't think of his name. It's Jack Numberg. That's who it was. I approached him with my theory, and I go, can you explain this? Why is it transmitting in American deer and American deer mice? And he started to pull out, the whole gaslighting card of aliens and whatnot. So it's got a little close to home for every scientist, I think, to basically, answer this question. Even people like Jack Numberg at the University of Montana, who entertained the idea that it was a Wuhan lab leak, are now going to basically poo-poo on the idea, because this thing's got a little too close to home for everyone.
Okay, so to follow that through, so first explain why they would use consensus genomes again. Consensus genomes meaning that the virologist, mainly Barrick, he's the only guy that I think I can name who's ever made a consensus genome, where you take 5 closely related viruses, pick the most popular nucleotides in certain areas, and you create a sixth virus that has it's very closely related to all of them, but none of them are exactly the same. What would be the purpose of doing that? We know he has done that, created a consensus genome. What's the purpose of it?
He wants to, well, we can only ask him, but he won't talk.
No. I mean the theoretical reason.
Yeah, I understand.
Yeah.
I want to make that point though.
Yeah.
Barrick has not talked since DARPA Diffuse leaked. He was talking. Now he hasn't. Now, if people want to defend someone that won't defend themselves, be my guest. But I mean, that alone is a great piece of evidence right there that the guy has gone radio silent since September of 2021. And he was open until then, but now he's gone silent. You know, he recently did a talk to Time Magazine, but who reads that anymore? And in that Time Magazine piece, he pointed at, the Montana lab, if he knew what he was looking, or what he was saying. But why did he, going back to the genome sequence, which is what most people want to study when it comes to lab leak, and it's only part of the puzzle, a very small piece of the puzzle. But inside that DARPA Diffuse proposal was a consensus sequence. That was the idea. What is that? You take the genome of a geographic area and you average it. And in the diffuse proposal, Barrick wants something like RATG13, Banal 52, RMYN02, the other viruses from Thailand and Cambodia to go into a giant mixer, a food mixer, and he wants it to come out and let none of them be more than 5% away. So that's essentially what we're finding, these natural, quote unquote, natural viruses found in bat caves that are just 5% off SARS-CoV-2.
So is that as part of the therapeutics to make that slightly different from the other ones, and that's where he would put on a new receptor or a new gene, like how we do spike protein with the mRNA. He would do the same thing with these to slip in with this consensus genome virus. It's a completely synthetic virus, not in nature, and he'll put in a gene that expresses a protein or a part of one of those other viruses to create immunity in the reservoir population. That's what I assumed. What was the purpose?
Yeah, to use his words, not mine, he wants to box in the virus. He calls it a bookend. If you listen to him closely, and I've listened to every word he's ever spoken and read every word he's ever written, he's looking to box in is this term. He calls it a bookend. It's kind of a neat little term, but like a bookend for people that don't read books anymore is the way he even puts it when he was up in front of Congress talking about it. These bookends basically from A to Z, and he wants to find A and he wants to find Z, and then he wants to create a barrier. So that barrier was essentially in his mind something that was 25% off SARS-1. SARS-CoV-2 was 22% off SARS-1. So he essentially created a bookend of a genome sequence that would box in this virus using his vaccine to prevent a jump of species in the future. So he's going to preempt the next zoonotic spillover event by creating a vaccine that is just on the other side of what could potentially jump species. And he's now boxing it in, like with a vice, basically, is my term, not his, but his term is a bookend. So I can only use his terminology and his mindset. But yes, it's very grandiose, no doubt about that, but when you can do grandiose things like create a synthetic genome from scratch, you can do grandiose things like that. So inside that, DARPA Diffuse proposal explains exactly what we've seen. He had a copy of RATG13 and Banal 52. Both of these viruses were collected with U.S. tax dollars. And the whole idea of, something like PREDICT, the USAID PREDICT program, was that you would predict the next spillover event. And you do this by going out there and harvesting all these bat samples. And with your tax dollars come strings attached. The people that collect these things have to kick back a copy to the U.S. virologist to play with them in their lab. And so that's essentially what we see. Every genome that's been closely found to SARS-CoV-2 has U.S. tax dollars attached to it, RATG13, Banal 52, RMYN02, every single one of them. So these genes are basically been harvested over time, and Barrick has come to this idea that he can box in the virus with this genome sequence, this beautiful, I call it his pietà. It was his masterpiece. This thing was beautiful. He did, again, no seum not to fool us lab leakers. It was to fool the animal's immune system. It was a beautiful place for it to leak, well, unfortunately, from Chinese labs with a Chinese bat virus. But it's one of the greatest ironies of life. But yes, that is essentially what he's done. He's created a genome sequence that was so great, so particular, so beautiful. It fooled all of us, every single one of us. It even fooled full-time virologists, as we've seen in January 2020. They're going at it, and give them credit. They're at least debating it. And they're having a hard time understanding it. The only thing that stuck out was, again, the fear and cleavage site. And flanking that fear and cleavage site is a BSAXI restriction site, which Barrick himself published in 2017, which he was asked about by the U.S. military in 2020, which he just completely just discarded and didn't even mention. So, my point to fellow lab leakers is that if we admit Barrick created the genome, he's the only person that could have created the genome. He said he would create the genome. He did create the genome. This story only gets better. I understand it's a lot to digest because a lot of people have this idea something bad happened, therefore someone bad did it. You must keep the word accident in the back of your mind, the front of your mind whenever you're going through this.
Yes, so to clarify a couple of points you made. Number one is if you know he's capable of creating a consensus genome, he has the reverse genetics. He's been doing this for 20, I think in 2003 he did a reverse genetics on SARS-1. He's been doing this for 20 years. He is the expert. He is god of coronavirus reverse genetics and genetic manipulation. No question about it in this field. And there's not a single person you will ask, could he have done it? And they'd go, well, of course he could have, but this happened in Wuhan. That's where this disconnect is happening. And here's where some of the pushback would be. Before, not now, but before, was that he didn't have these genomes. To come up with the consensus virus, you'd need to have full-length genomes for those 5 viruses, or however many viruses he's using to come up with that consensus genome. He'd need to have the full 30,000 kilobase gene sequence. But what you just said, and what I read, again, I reread the DARPA proposal after reading your work, and in there, Peter Daszak, EcoHealth Alliance, the guy who actually did this diffuse proposal, he was actually the principal investigator with EcoHealth Alliance. He said in there that he has this paragraph talking about, we have all these data sequences from all of our subcontractors from previous things that they've been doing, which indicates to me, number one, you said Ralph Barrick has it, and so does EcoHealth Alliance. And so when we talk about solving the mystery of the origin of COVID, we keep saying never is going to happen because Wuhan won't give us their data. It's irrelevant. We have all that data. There's like duplicate and triplicate copies of it. We have it probably in the NIAID. We definitely have it at the University, most likely have it at the University of North Carolina, because he was one of the people that what do you call them? Shared trade agreements or material...
MTAs, material transfer agreements, yes.
MTAs would dictate that any virus they had sequenced in Wuhan, EcoHealth Alliance, Ralph Barrick, and probably the NIAID, and who knows else might have it. But they have this data somewhere, unless what I worry about is what you said, is somebody burned all the data, but they should have all this data somewhere. If you just said, let's look at all the sequences you've worked with any coronavirus research in the last 3 years.
Yeah. Well, if you look closely, Frank, in 2018 and 2017, they published RATG13. Now, this is where it really gets hard on some people's brain, including mine. But once you accept the hypothesis that only Barrick is the only person on the planet that could have created it, and he literally said he would create it in 2018, you start looking at his files very closely in 2017 and 2018. And in 2017, he published a cDNA reverse genetic system that could create a new virus from scratch, essentially a seamless virus, a scarless virus from scratch. And in there, he talks about a type 2G restriction site called BSAXI, which he used to, again, flank his fear and cleavage site, which, again, UNC published in 2018. They published RRAR, which is the fear and cleavage site, in 2018. It gets better than that. They also published SVAS, if I have this right,
yes,
essentially. Mm-hmm. And that is a very unique sequence inside RATG13. It's not found in any other genome. And they published that in 2018. So they are fiddling with this thing in 2018. She has shared the sequence with Ralph Barrick's lab, just like she did in 2013. She of the Wuhan Institute of Virology shared what's called SHC-014 with Ralph Barick's lab. Ralph Barick sat on it until 2015. Then when Ralph Barick published it in Nature, which is what they're all trying to get into, a Nature paper, it's their business card, essentially, he put Shizeng Li as a co-author on the 2015 paper simply because she shared the sequences before they were published. And that's what Ralph Barick said in his own words in that June 2021 interview, because that's what threw off everyone. Why is Shizeng Li on this 2015 paper showing a SARS-like virus that could be created in a lab? And it turns out she had nothing to do with it. It was Ralph Barick's work. She just shared the sequence. So SHC-014, if you search for that on Twitter.
Oh, no, that's the one that Fauci is up all night on January 31st.
And this idea that, Fauci's told at midnight on Friday or 10 p.m. on Friday by Christian Anderson.
Yeah, he was up till 3 in the morning, yeah.
He's told it's engineered, and he's up until 3 a.m. covering his tracks. And people are like, he's covering for China. No, these are selfish people. They're covering for their own hide.
So you made a really really crucial points that again that I was not aware of this myself, and I've been looking at this for 3 years. It's very important to note that Ralph Barick says, as do all the other really high-end synthetic virologists like Drosten. He's at the top of the food chain as well in reverse genetics. He said this couldn't be done in the Wuhan Institute of Virology. They don't have the capability. When you have an expert like Drosten and you have an expert like Barick both saying they don't have the technology to do this kind of stuff, we do that all here in America. When he says that, it's like, I go like, but how about that? You just told me the SHC 014 study. When I read that, I thought, oh, that means Shizeng Li could have done this, because there she is doing chimeric virus researches, learning all the tradecraft from Ralph Barick. So she knows all that, and now she dumps it over to the military arm where they start tweaking it around in their own lab. But then when we find out later on, Barick says, oh, I never gave her that technology. I never did any of that stuff with her. All she did, she gave me the sequence, which he was able to resynthesize the virus just from the 30 kilobase sequence.
Yeah, that wasn't even 30 kilobase. He was given the spike.
Oh,
and
okay.
He turned it into a 30. The spike is like, I don't know, it's like 8,000 nucleotide, and the whole genome sequence is 30,000. So yes, he literally created something from nothing. Well,
that was that consensus genome is a completely novel virus.
He's putting on the training wheels for himself, and this idea that he shared his technology with he has a $100 million lab. She has a $2 million lab, as best I can tell. He has, all the latest and greatest, software. He has what's called the Illumina machine, the latest and greatest. Shizeng Li didn't even own an Illumina machine to sequence these viruses until 2018. She bought a used one. It's a community one. That's why you get all these kind of contamination research studies by lab leakers. It's a community. It's like a community water fountain. It's basically got all this cross infection on it and all this other stuff. So, when you look deeply into it, she has a lab that has a lot of people. It's like 30 people, which is about the same number as Ralph Barick has. But Ralph Barick's lab cost a lot more than her lab. So 30 Westerners is a lot more than 30, Chinese, people working in a lab. So it's not an apples to apples comparison. But yes, that is the leading theory, is that if Ralph is guilty, he shared his technology with her. You don't share your $100 million biotech with anyone. And, it goes back to something I tried to reference out of many references. You know more than you can tell.
Mm-hmm. Oh, yeah. So on that, I think that clears up that question mark. That's like, I do believe Drosten does have skin in the game, but when he keeps saying this could only be done in America, and when he eliminated, I think he was genuine when he says couldn't have been done in the Wuhan Institute of Virology, and I do believe them now. A lot of these guys were actually telling the truth, but like you said, a partial truth. They didn't say this could have been synthesized, could have been engineered. Absolutely, just couldn't have been engineered there. Therefore, I don't believe it happened because it leaked over there. So where we've been alluding to is where the rubber hit the road was you actually have mapped out a very plausible, based on a lot of information, pathway where it got from Barick's lab with this new CREED proposal that you had just mentioned. There's this guy in the Rocky Mountain Labs. He's actually, and this is what convinced me of it, I didn't know this, the sixth Munster, Vincent Munster, was a disciple of Ron Fouché and Marion Kupmans. Ron Fouché is the guy who did, I've described this in the show before, the ferret, the aerosolization of H5N1 that freaked everybody out in 2012 or 11 or 12, caused the moratorium when you aerosolize H5N1. It has a 60% reported case fatality rate. If this thing gets out of the lab and it's aerosolized, I mean, think of what would happen if SARS-CoV-2 had a 60% mortality or case fatality rate. It'd be 1 billion people dead. And this guy's a disciple of his, Vincent Munster. That's not a criticism. I'm just saying the guy has capability, has had a lot of experience in a lab where they created potentially the most dangerous pathogen, aerosolized pathogen in the world. Thank the Lord, it never got out. This is the gentleman who Fauci hired at the Rocky Mountain Laboratory in Hamilton. So the idea is to make this a scalable immune modulator, this vaccine that I'll just call it infectious vaccine for the bats in the caves in Wuhan. So that's the goal, to get it to those caves in Wuhan, immunize all those mice, all those bats in the cave, and get rid of SARS, all these SARS coronavirus, make the bats immune to SARS. So Ralph's figured out how to create that live attenuated vaccine against them, but it's spreadable. It can spread from bat to bat or human to human or white-tailed deer to white-tailed deer. But he doesn't have the operation where he makes it quote-unquote aerosolized or the scalable project. Munster's in charge of figuring out how to take that virus and infect all those bats in the cave in a cost-effective manner. Do we have any knowledge on what